Ladies and gentlemen, welcome to the Distinguished Speaker Series. My name is Marco Bassetto, and I am a postdoctoral fellow in professional pleasure to introduce our speaker of the day, Dr. Thomas Michael Redmond. Dr. Redmond is a senior investigator.
We can't hear you very well.
Can you speak up or move the microphone closer to your mouth? Lift it up a little bit.
Is it good like this?
I don't know, can somebody increase the volume? Is there a volume control right there at the microphone?
Microphone is off.
Okay, here I am. Can you hear me? Yeah, so I am Marco Bassetto, and here we are at CTVR. Welcome to everybody. Today I'm going to introduce Michael Redmond. So, as I was saying, the contribution of Dr. Redmond to vision research are multiple. Indeed, we live in the era of gene therapy, where it is possible to treat RPE65-related Leber congenital amaurosis, and Dr. Redmond contributed to this success by adding several crucial pieces to the puzzle. One day, Dr. Redmond set off for a scientific journey, which began with this statement. Recently, some RPE-specific monoclonal antibodies have been described. Protein recognized by these antibodies, because of their specificity for the RPE, may participate in certain RPE specialized function. With his colleagues, he cloned the gene corresponding to this protein and named it RPE65. Back then, he clarified, no human disease has yet been shown to result from a mutation in a gene required for RPE function. In the next 2 years, Dr. Redmond published two important works, among others. The first that links the RPE65 gene to blinding disease with early onset of retinal degeneration. In the second, where he is the first author, he describes the RPE65 knockout model. In this paper, you find a distillate of the knowledge about the visual cycle gathered up to that point in time. Here, Dr. Redmond concluded, 1. RPE65 may be the effector enzyme in the isomerization step yielding 11-cis-retinol. 2. RPE cells take up both adenovirus and adeno-associated viruses, thus allowing the possibility for rescue experiments. And 3, co-ERG function in the RPE65 deficient mice is preserved. The first two conclusions have been proved true. About the third one, well, there have been some surprises, but this is a different story. Dr. Redmond produced many other important pieces of work, but there is one of them that deserves our attention because it is connected with a beautiful story. There were two heavyweights in vision science that were competing for describing the enzymatic mechanism of all transretinal isomerization to 11-cis-retinol. These labs were publishing work supporting the respective hypotheses. But then a young blonde PhD student solved the structure of RPE65. 1 year later, Dr. Redmond first authored a paper that demonstrated once for all that the isomerization of all transretinal to 11-cis isoform does not involve a nucleophilic attack of RPE65 to the vitamin A, but more plausibly, it involves a carbocation intermediate. There are, of course, many other stories, anecdotes, academic achievements, and honors that should be mentioned here, but at this point, I do not subtract more time to our speaker. Thus, without any further ado, I want to invite you to follow Dr. Redmond's lecture, which is entitled Squaring the Circle, the Missing Bits of RPE65.
Thank you very much for the very exceedingly kind introduction. Today, I want to talk about some of the missing bits of RPE65. And I just want to echo something that Bob Mulday said 2 weeks ago, and that is 11-cis-retinal is the most important molecule in the eye, and in its absence, nothing else in the eye matters. 11-cis-retinal acts as a photon trap, allowing perception of visual environment, and without 11-cis-retinal, vision as we know it would be impossible. So where did this all begin? Well, we can go back to the Burgess Shale 500 million years ago. This critter here that oops. That bears a resemblance to the
You're good, Mike.
yeah, I'm looking for the
Pointer?
pointer. Yeah, thought I had it, but in any case, this creature, which bears a resemblance to lampreys, has been found in the Burgess Shale, and evolutionarily, we believe that the vertebrate visual system evolved with the last common ancestor of jawed and jawless vertebrates. So why the vertebrate visual cycle and why RP65? So like I said, the vertebrate retina was well established in the most primitive vertebrates. Cones and rods are also present. However, for this vertebrate retina, constant production of 11-cis retinal is required because of the retinal of vertebrate opsons being monostable, unlike invertebrate opsons, which are bistable. You can, after the photon capture in a bistable, you can absorb a second photon and regenerate the opsin in situ, but monostable opsons release their photoisomerized all-trans retinal, and thus the opsin must then bind another 11-cis retinal to return to the light-sensitive ground state. And to accommodate this process, the earliest vertebrates had already evolved an enzymatic mechanism called the visual cycle to permit this conversion of all trans retinoids back to 11-cis retinoids in the dark. And this acquisition probably allowed these earliest vertebrates, such as lampreys, to colonize darker environments. However, neither RP65 nor LRAD are found in primitive chordates or invertebrates. But both of these had evolved by the last common ancestor of the jawed and jawless vertebrates. And RP65 evolved from ancestral carotenoid oxygenase enzymes, and at the same time, concurrently, to generate the substrate for RP65, LRAT enzymes evolved from the N1PC P60 family. But RP65 is a seemingly radical departure from other carotenoid oxygenases, as it neither uses carotenoids as substrate, nor does it oxidatively cleave carbon-carbon double bonds. Instead, it does a no-alkyl cleavage of the retinyl ester, and this is quite with the isomerization of the retinoid moiety, quite a unique mechanism found nowhere else in biology. So we've known the bones of the visual cycle since almost 90 years ago, when George Wald published this diagram, this paper in Nature. And the visual cycle was described as a process whereby visual pigment sensitivity is regenerated following light exposure. Today, we have been, in that time, this 80 odd years since then, we have been putting together this current concept of the visual cycle. And I won't say too much more about this except to point out LRAT and RP65 in the process. And as we all know, the visual cycle is prone to numerous sites of disruption by genetic mutation. RP65, like I mentioned, has been around conservatively 500 million years. We find it in the lamprey, where RP65 is actually quite abundant in what passes for the lamprey RPE, as well as being quite abundant in the human RPE. And as was alluded to earlier, it's been shown that it is essential for the production of 11-cis retinoids in the visual cycle. Also, over the years, when from its first evolution, it has changed remarkably little. The lamprey to mammalian RP65 shows a 72% identity and an overall 92% similarity. That's not too much change over the course of 500 million years. And as you can see, there are regions where there is a significant level of identity, complete identity in the structure, in the sequence between all the different RP65s from mammalian down to cyclostome. So today's talk, I want to talk about two things: the functional role of RP65 palmitulation, and also the structural context of the palmitulation at cysteine 112 and its role in RP65 endoplasmic reticulum membrane. So the majority of the work that I'll talk about was done in my lab by a very hardworking, very talented postdoc research fellow, Sheetal Uppal, with the important assistance of the staff scientist, Dr. Eugenia Polyakov. In addition to several other members of the lab. One person I should call out is Dr. Ting Liu, who began the project before she returned to China several years ago. So the palmitulation of RP65 has had a, I won't say long, but it certainly had a tortured history. Back in 2004, Xu et al. In the Rando lab advanced palmitulation switch mechanism, proposing that RP65 was a shuttle that carried retinyl esters to the, at that point, insufficiently identified isomeral hydrolase. In 2009, Takashi et al. From Jay Ma's lab and Philip Kaiser et al. Using mass spec separately identified cysteine 112 as the palmitulation site of RP65. However, in 2010, Yuan et al. In Travis's lab, Gabe Travis's lab, also using mass spec, concluded that RP65 was not palmitoylated and that RP65 membrane association occurred via electrostatic interactions. And below at the bottom here shows a few different models taken from Philip Kaiser's review in progress in retinal and eye research, showing A is the electrostatic model, B would be the palmitulation model, showing 3 palmitoyl groups, and C, monotopic peripheral membrane association, and D, an association with a membrane inserted other protein. So I need to describe more closely the palmitulation switch mechanism, as this will come up a little bit later in the talk. So the palmitulation switch mechanism proposed that RP65 is alternately a chaperone for retinols on soluble SRPE65 in the dark, and for retinal palmitates on the membrane form in the light. And to affect the so-called switch, LRAT palmitoylates SRP65 to MRPE65. And then this triply palmitoylated MRP65 shuttles the all-trans retinal palmitate to the isomeral hydrolase, and then donates the palmitoyl moieties to LRAT to esterify 11-cis retinol. There are many issues with this mechanism, and the major drawback was that RB65 is actually the isomerase and not its chaperone for its substrate. And another issue was to detect the palmitulation. Xu et al. Used tritiated palmitic acid labeling, and this is a method that's prone to artifactual labeling. And finally, none of the identified cysteines actually are functionally palmitoylated in RP65. But despite these issues, we ask the question, are there kernels of truth in the model? So we wish to check this out and to establish what is the story here. So we used more robust detection of palmitulation using methods ASOL-RAC, resin-assisted capture, and acyl biotin exchange, and used exhaustive site-directed mutation to map sites and to analyze the role of palmitulation. So this is a description of the method that Sheetal employed, and it involves blocking thiols in the sorry, I'm just trying to, yeah, it involves blocking thiols in the it involves blocking free thiols with NMTS, and with these blocked to remove palmitates using to remove S-palmitoylated palmitates, cysteine palmitulation using hydroxylamine, and then to either replace these with biotin, or to use the free palmitil, the free thiols to be pulled down on thiopropyl cephalose beads. The long and the short of it is that RP65 is palmitilated, and in panel A, you can see the results from both the ACEL-RAC and the ABE, showing palmitulation of the molecule. In B, we have a positive control using rhodopsin, which is structurally palmitilated, as we all know, and a negative control, CRALBP CRALB, which is not palmitilated. However, we found that only about 25% of RP65 is actually palmitilated. We did site-directed mutagenesis to narrow down all of the all of the cysteines that are present on RP65 down to 3 remaining candidates, and of these we identified cysteine 112 and cysteine 146 as being palmitilated, the sole palmitilated residues on RP65. Very important finding was that RP65, the palmitilation, is modulated by the activity of LRAT. There. Sorry about this. I'm trying to get the pointer to work, but the pointer seems to be missing in action today. The point being, RP65 alone by itself is palmitilated, but in the presence of active LRAT, in the presence of retinol in panel, the left side of panel A, the center lanes showing presence of retinol, the palmitilation of RP65 is abolished. In the presence of inactive LRAT, the cysteine 161S mutant, palmitilation is restored in the presence or absence of retinol. And also we identified that palmitilation is required for the association of RP65 with membrane by comparing it with 2 other proteins, one that is palmitolated PhD 95 and one that is not, RDH5, and we can see that hydroxylamine treatment is able to shift the RP65 to the soluble phase, which is indicates removal of RP65 from membrane. We also were able to show that inhibitors of palmitulation also reduce RP65 palmitulation. Furthermore, that C112 is the one cysteine which is required for RP65 membrane association. I'm having a problem with the controls here. And we also were able to find that BCO2, which is another member of the carotenoid oxygenase family, of which RP65 is a member, is also cysteine palmitolated. But what's interesting is that the palmitulation of BCO2 is lost in the absence of, in the presence of its xanthophyll substrates, and this is maybe an interesting parallel with RP65, seeing that RP65 may also lose, you could construe that RP65 loses its palmitulation in the presence of retinyl palmitate by virtue of the LRAT activity. So the second part of the talk is to put this cysteine 112 palmitulation in a structural context, and to show how it is related to the association of RP65 with ER membrane. And this also is features the same cast of characters, Sheetal and Eugenia and Ting Ting. So our question was, what is the structural context of C112? And there's a major problem here, and that is that cysteine 112 is located in a region that is part of a crystallographically unresolved region in RPE65 structures. These are all structures that have been done and accumulated by Philip Kaiser and the Palczewski lab, and this is seen multiple publications from these groups. And the region where C112 is located is lacking in resolution, which is, I would say, a severe problem for for such an important part of RP65. And when we look at RP65 in this region, we see that in actual fact, it is very highly conserved, all the way back to hagfish. We know that sea lamprey has a functional RP65. It can actually make 11-cis retinol. Not so sure about hagfish RP65, but this region has been conserved to a great extent over the last half billion years. However, when we first cloned RP65, Christian Hamill and colleagues, we showed that that it had an amphipathic helix character, and we have done alanine screening of the region, and we find that it's important for RPE65 membrane binding, some more than others. Also, the mutation of residues within this region affects RPE65 palmitulation status. Many of these mutants of this region are simply not palmitolated. Also, it has a mutation of these residues has an adverse effect on RP65 isomerization activity. And it's a hotspot for missense mutation. So it appears to be, this appears to be a really important part of RP65 real estate. This 7 out of 19 residues is above the odds. So we decided to look closer at this region, and as I indicated earlier, RP65 forms an amphipathic alpha helix, which is actually of really high, large hydrophobic moment. You can see in the bottom panel, panel A shows a stockade, as it were, of phenylalanine phenyl rings at the hydrophobic face of the amphipathic helix. So we wanted to look closer at this, so we chemically synthesized 2 peptides corresponding to this region, one which was unmodified, and the other was modified by C112 palmitolating cysteine 112. And we use these to analyze the behavior in different solvents and in binding to membrane mimetics using far UV CD spectroscopy, circular dichroism. spectroscopy and did further analysis on behavior in detergents. So in comparing the two peptides, I want to draw your attention to the red highlighted numbers for alpha helix that in the presence of either water or buffer, that the non-palmitoylated peptide had little alpha helical structure. However, in the presence of 50% trifluoroethanol, which stabilizes alpha helical structure, this went up to 95%. However, the palmitoylated peptide, palmitoylated at C112, was showing alpha helical character no matter what the buffer. And this was borne out in these pictures. This is borne out in these pictures of the far UV spectra, showing in the non-palmitoylated little structure in either buffer or water, but formation of alpha helix in the presence of TFA, whereas the palmitoylated peptide is has significant alpha helical structure right out of the box, and in the presence of 50% TFE, this structure is even more stabilized. And similarly, in the presence of detergents, both the non-palmitoylated and the palmitoylated also obtained or also had alpha helical character, which again is more stabilized in detergents in the palmitoylated, both the palmitoylated and the non-palmitoylated. We looked at how the structure, alpha helical structure, is stabilized in the presence or absence of detergent micelles, and in the non-palmitoylated peptides on the left side, in the presence of detergent, it had a good chromatic separation compared to when in the lower left panel, where the detergent micelles were not present. The detergent micelles, the absence of detergent micelles caused the peptide to actually interact with the chromatographic medium and media, and so it was significantly delayed in its elution. That was less the case with the palmitoylated peptide, which seemed to be able to elute quicker, and these are borne out in the graph shown below. So we then asked the question. We then asked the question whether this peptide, this 107 to 125 peptide, was sufficient to target RP65 to membrane. So we did a couple of experiments to test this targeting property of targeting properties of the RP65 AH105 125 region. So we made two constructs, a maltose binding protein construct, two fusion constructs to determine if the sequence can target it to liposomes, and in addition, a green fluorescent protein fusion to see if it's capable of actual subcellular targeting. So for the maltose binding protein, in panel A, we see purification of a maltose binding protein and a maltose binding protein AH107-125 fusion construct, and you can see the slight increase in size expected in the fusion protein. This we mixed with NBD PC and DOPC liposomes in a ultracentrifugation experiment where the proteins, either the maltose binding protein alone or the fusion, were mixed with 30% sucrose and placed at the bottom of a tube and centrifuged at 170,000 G for 40 minutes, and after the centrifugation, the liposomes had shifted to the top and were at the top of the gradient. We analyzed this by SDS page and Western blotting, and we found that the maltose binding protein shown in the graph on the far right, that the fusion protein was found in the top layer more so than the non-fusion construct. So looking at the green fluorescent protein, we transfected the GFP construct into COS7 cells, and with the unfused regular GFP, we see in the top set of panels, we see the GFP is doing its usual nucleophilic nuclear localization of protein, whereas the fusion AH107-125 fusion actually pulls the GFP out of the nucleus and into the cytoplasmic compartment of the cells. So to investigate this a little bit further, we used the DS-RED endoplasmic reticulum marker that is a marker for ER membranes, and in the presence of GFP alone, we see that the GFP stays with the nucleus, but in the case of the fusion, the 107 to 125 fusion, that this pulls the GFP out of the nucleus, and is found to co-localize with the GFP marker. And then separately at the bottom panels, this shows RP65 expression of an RP65 construct, and this is quite similar. In the far right, the images in the merged lanes are quite similar between the anthopathic helix fusion and the RP65 co-localization with endoplasmic reticulum. So we also did molecular dynamics simulation of this region to show interaction with protein, with a model membrane, and this is showing the peptide dipping into the membrane and interacting fairly deep into the membrane. And I will stop this and just go back to showing the graph of the interaction, showing at the early stages on the bottom far left, followed by association. Hello? Followed by association with the membrane, and subsequent to that, total burial in the membrane. So we are suggesting that this is an example of a membrane sensor, and that this is the structural rationale behind RP65's interaction with the endoplasmic reticulum. And there are examples from previous work in other proteins, one of which is PIP5K, which utilizes the canonical activation loop of PIPKs to and basically makes it do double duty as a membrane sensor. And then also the RDH peripherin RDS, peripherin 2 RDS of rod photoreceptors, which uses an anthopathic helix to modulate curvature. And so just to summarize, we have a proposed role for this 107 to 125 in RP65, and we hypothesize that in RP65 in the soluble form that 107 to 125 is intrinsically disordered region and potential membrane targeting motif. And then it's capable of forming an amphipathic helix upon interaction with membranes. And that this IDR alpha helix, amphipathic helix structural transition is key to the role of RP65 in the visual function, in the visual cycle. And the reason for this is that IDRs, for the most part, lack defined secondary, tertiary structures. There's no motif, there's no sequence that says this is an IDR. But these disparate sequences can transition from disordered to ordered conformation upon binding to proteins or membranes, and on the basis of this, 105 to 125 transition allows phase separation between a membrane unbound and bound states, and also the duality would explain both the observed lack of structure in crystals, by large apparent D factors, and the prediction of highly amphipathic helix. So another feature is that the sequence also has a large moment, hydrophobic moment. This is a feature of surface active antimicrobial peptides, which burrow deeply into membrane. So would indicate that it would be allow this region to deeply penetrate into the bilayer to access retinal esters, which may be located quite deep in the membrane. And also another intriguing aspect of this is that palmitulation further enhances formation. We find that the unmodified peptide can form an amphipathic helix in the presence of membrane mimetics. The amphipathic helix formation is automatic in the palmitulation, palmitoylated peptide, suggesting a direct local conformational switch. And it's also quite a novel finding, since we have not been able to see it in any other protein in the literature. And we asked the question, is this the identity of what I mentioned before, the palmitulation switch? And finally, we find that the amphipathic helix sequence, the 107 to 125 sequence alone by itself is necessary and sufficient to target heterologous proteins to the ER. And I just want to present this model, and I will skip over the conclusions and give the most important slide of acknowledgements. And the folks in red have contributed directly to this to the research presented here today, also including some alumni. I mentioned Ting Ting, and we also have the BCO2 work was done in collaboration with Eugene Pinkasik's lab in Yukon, Connecticut. And with that, thank you for your attention, and happy to answer any questions.
This is Dorota Skowronska-Krawczyk from Center of Translational Vision Research. Wonderful talk and really fascinating. I have two sets of questions. One is about the palmitulation. Do you know if palmitulated RPE65 is organized in subdomains in ER membranes, or it is just dispersed? And if localized, which is usually the case for lipid modified proteins, did you try to look at the composition of such subdomain? And the second question is it always palmityl group? Did you find other fatty acids, and can it be unsaturated and not always saturated? Thank you.
Thank you. Yeah, the first question, in response to the first question, is we simply don't know, but that is an avenue that we are actively involved in trying to do. I had a slide that I didn't show of the kind of history of RPE65 studies in the last 30 years, and I think that is, that would correspond to the last area that needs to be still needs to be covered. We've done a lot of work on the biochemistry, all the different labs that have worked on RP65 over the years, on the biochemistry, molecular biology, molecular genetics, structure, catalytic mechanisms, etc. But we haven't yet gotten to I think what you might want to call the protein dynamics of RP65, and also RP65 in the greater context of the visual cycle. And it's been a kind of a term that was bandied around in the last 10, 20 years by several workers in the area of visual cycle, of a visual cycle complex, and that's something that we're really trying to get at in our research, upcoming research. As to the second part, I think it is at least theoretically feasible that you could have other acyl groups besides palmityl modifying RP65. We haven't looked directly at that, but we would not be surprised.
Steve. So, Michael, beautiful work. I wanted to just ask a question, maybe to see if I understood this correctly. So there are, did you say 3 or 4 potential consensus sites for palmitulation, but only one, the 112, is utilized? Is that correct?
There's 3 potential sites that we have found. C112, 145, 146, and 195. And of these, we have focused most on C112 because of its importance in targeting. The C145 is we're less clear on what that that might be doing, but that's not involved in membrane association at all, because
Okay, I understood that part. I thought I caught you say that only a percentage of the molecules are actually palmitoylated.
yes.
So what's the basis of selectivity of how does
Well,
the cell sort that?
Yeah, we our concept is that it's a concept of kind of dynamic palmitulation. We kind of compare that to rhodopsin, which is, as you know, structurally palmitoylated. And the palmitoyl groups play an important role in stabilization of rhodopsin in the photoreceptor outer segment membrane. So that the discs that's important for rhodopsin. But in the case of RP65, we think that there is somewhat of a dynamic palmitulation, and we're trying to figure out how and why in work that's already underway. We are looking at the possibility that there are specific Z-D8HC PATs that may specifically palmitulate RP65. So we're looking at that in the context of RPE. And as you know, I think there's like 22 of these, so we have to narrow down what it might be.
Yeah, and finally, that last group of experiments where you showed that the amphipathic helix 107 to 125 was necessary and sufficient for membrane targeting is a beautiful set of experiments. A finesse experiment that people who use that trick do is to scramble the peptide to show that the scrambled form doesn't target. Did you do that?
Yeah, no, we haven't done that. That's a valuable suggestion.
I don't think your story is any diminished at all. It's just a finesse, I think.
Yeah. Well, I think doing that kind of scrambling experiment would mess up the amphipathic helix.
That's the caveat.
Yeah,
yes.
Yeah. But I think that is a positive and a negative.
Good. Thank you so much, Michael.
Sure.
John? Hello, Dr. Redmond. Thank you for this wonderful presentation. I was wondering about the role of LRAT activity in decreasing the amount of palmitoylations in RP65. Maybe you've already elaborated on this, but maybe I just want to get maybe a clarification. So does the depalmitation of RP65 by LRAT activity decrease the association of the RP65 from the membrane? And my second question is, what is the, I guess, physiological purpose of depalmitation of RP65 in the context of the visual cycle, given that it's immediately after LRAT?
Yeah, these are crucial, I think, questions getting at the physiological role of RP65 and LRAT. As you no doubt know LRAT and RP65 are obligately tied together. RP65 is dead in the water without LRAT, in the intact animal. RP65 is dead in the water without LRAT. And the phenotype of an LRAT knockout is quite similar to that of RP65, no vision. We have speculated that this could be evidence of RP65 being a palmitoyl donor to LRAT, because LRAT normally takes the palmitoyl group from lecithin, from phosphatidylcholine. So the first position on phosphatidylcholine would be used by LRAT to transfer. LRAT essentially self-palmitoylates and then transfers that palmitoyl group to retinol to make the all-trans retinol palmitate. And it could be that RP65 can actually donate that that palmitoyl to LRAT directly, and that would also tie in with the Xu et al. Palmitoyl switch mechanism. But we don't know. We don't have any direct evidence beyond that experiment to really say yes or no on that.
Vladimir, Alex, and final word goes to Professor Kaiser.
Hey, Mike, thank you for the talk. And I think John, in a way, asked my question, but very broadly speaking, is the activity of RP65 different in darkness and in light in vivo in the RP cells?
That's a very, very good question to which I have no answer. I'm sorry. I would imagine, experiments have been done by other labs to indicate that there is a light dark cycle, but whether there is a catalytic difference or a mechanistic difference in RP65 in light and dark, I don't know.
Hi, Dr. Redmond. This is Alex from Dr. Podczewski lab. Maybe I have missed it. I was just wondering whether this C112, or in more broadly speaking, this alpha helical structure is some known site of human mutations that cause some vision impairment phenotype, like leading to LCA or UN.
I showed in my graphic on that region being a hotspot, that there is actually in the Leiden database, LOVD database, that there is actually C112Y mutation that that is pathogenic or at the very, very least a variant of unknown significance of the US. But it is known that a mutation at that site is known.
Thank you.
And the last question goes to Philip.
Hi, Philip.
Hey, Michael.
Hey, Michael.
Hi.
Fantastic talk. I really enjoyed it. So one just sort of minor question. Did you try in your detergent experiments with your peptide? Well, you showed that that this peptide will adopt an alpha helix in the presence of membrane mimetics like DDM and even SDS. I was curious if you tried another like non-ionic detergent like C84 or C86, since that's what RP65 is crystallized in. It'd be kind of interesting to see what the impact of that detergent, that type of detergent is on the structure of that peptide, and might help explain why we have difficulty resolving that in the RP65 crystal structures. So that's that. And then just kind of a comment, do you think, there's this interesting trafficking problem with RP65 where you've got kind of a no transmembrane domain, you have to somehow traffic this to the ER membrane. And I've always kind of wondered whether this palmitulation, kind of as you're showing here, the palmitulation converts that sequence to an alpha helix to make it amphipathic, and then it can maybe dock on the membrane, and you could get rid of the palmitulation and it maybe stay put because you have the membrane there. So do you think that maybe the palmitulation is important for sort of initial trafficking of RP65 to the endoplasmic reticulum membrane?
Yeah, to answer your first question, yeah, we haven't tried that one, but I would imagine that at least for the non-palmitoylated peptide, that it would. And then for the for your second question, yeah, I mean, it sort of gets back to the earlier question regarding LRAT. We don't know where the palmityl group, whether it can be taken off by a depalmitylating enzyme, whether it's because palmitylation is actually quite labile. It can be it just goes by kind of a chemical, for want of a better word, a chemical depalmitylation, whether it's enzymatic or whether it's a donor kind of situation, giving it to LRAT. But I would imagine that and these are aspects of the protein dynamics that I alluded to that need to be clarified, we're headed in that direction.
All right, thank you, Dr. Redmond, for sharing your research with us. It was wonderful, inspiring. Thank you all, and see you next week.